All right, we're carrying on, and we are talking about nucleic acids now. Oh, wow. Nucleic acids. So, again, we've talked a little bit about what elements we find in the different biological molecules. Oh. So we've talked about carbs. We've talked about lipids. We've talked about proteins. Carbohydrates have carbon, hydrogen, oxygen. Lipids also have carbon, hydrogen, oxygen. Proteins have carbon, hydrogen, oxygen, nitrogen, and some sulfur. And then nucleic acids have carbon, hydrogen, oxygen, nitrogen, phosphorus. So the way to remember this is cho, chon, chomp. Cho, chon, chomp. And you just have to remember that this is the order here. Cho, chon, chomp. Cho, carbs, cho, lipids, chon or chons, proteins, and chomp is nucleic acids. You can also remember that nucleic acids have, nope, never mind. I'm gonna take that back. I'm not gonna finish that sentence. Would have been confusing. Right, so the monomer for nucleic acids is called the nucleotide, and if we have lots of nucleotides together, then we call that a polynucleotide. One of the things that I should just add is that there is the chemical bond that holds them together is called this is a long word here, phosphodiester bond. It's called a phosphodiester bond. So right here, these are all the nucleotides here. So these are the nucleotides, okay? And the covalent bond that's holding them together right here, this is the phosphodiester bond. Okay, we'll take a closer look at that in just a minute. So again, just like we said with proteins, if you have one nucleotide, then it's called it's just called a nucleotide. If you have two nucleotides, then it's called a dinucleotide. If you have three nucleotides, it equals a trinucleotide. And if you have many, then it's a polynucleotide. The two main examples of nucleotides that you need to know are RNA and DNA, and you should be able to compare and contrast the differences, which we will talk about in a minute. Now the other, let's talk a little bit about the functions of these things. So probably the most important part of nucleic acids, the most essential part, is that they store our genetic information. Okay, so they store our genetic information in DNA, and they also transmit our genetic information. Okay, so not only do they store it, they also transmit. And we should really put it here. RNA transmits our genetic information. Transmit is just another word for send. So if you transmit an email, you send an email. Okay, so there's 3 types of RNA that we need to know. There's 3 types of RNA. RNA, 3 types. The first one is called mRNA, and it's the messenger RNA. There is there is tRNA. This is called the transfer RNA. It's kind of like the decoder. And then there's ribosomal RNA. Maybe we should do this. Let's do this. There we go. I don't know why I didn't do that. That'll make a lot more sense. And then there's ribosomal rRNA, and this is ribosomal RNA. And this is the one that it builds ribosomes. And we'll learn that ribosomes are little protein factories, protein building factories. So the mRNA, this is the message. The tRNA is the decoder, and the rRNA is the protein building factory, essentially. Okay? So here you can see all of those things happening. So up here, we've got, this here is the DNA. Oops. So here we've got this twisting little shape here. This here is the DNA. So all of our genetic information is stored in here. So all of the things that give us like what we look like, how we function, all that information is stored in our DNA. And then that DNA, our cells are able to make a message out of that. We don't need all the DNA all the time. We just need pieces of that DNA. And so this mRNA just takes what we need from the DNA. It makes a little message. It writes a little message from the DNA. It uses the DNA, kind of like we go and find information in a textbook, and then we write it down. The textbook is the DNA. The mRNA is your note. Right? So here's the little message. That'll go out into the cell. And then here we've got this machine right here is called the ribosome. And this is the little protein making factory. And then these things right here, these are the tRNAs, the transfer RNAs. So these are the decoder. And you can see look what it's bringing, an amino acid. So it's changing the message from a nucleic acid to a protein. So RNA really is the bridge. RNA is like the bridge between, let's make it a different blue. RNA is the bridge between nucleic acids and proteins. And what you really need to understand is that our genetic information is stored in our DNA. Our cells will then change that DNA into RNA. RNA will transmit that message, that code, that information, and it will make, our cells will then make proteins. This is our trait. Okay, these are our genes, and the proteins are our traits. And RNA is kind of like the translator. Right, you can think of these as two different languages. Maybe this is Korean, and this is English. And this here is Google Translate. Okay, and we go from our genetic information, our DNA, where it's stored in the nucleus. It gets changed, it gets sent out as a message and changed into protein using RNA. And the protein is our trait. Because remember, proteins are structures, they're antibodies, they are enzymes, so lots of different types of proteins. All right, you need to know the difference between RNA and DNA. RNA is typically single-stranded, so you can see here that it's just one strand, it's just one string. Essentially, a strand is a string, right, or one piece. But DNA has two pieces. Here's one piece here, okay, on this side. Here's one piece. And then here's another piece, and they're connected in the middle. They're connected in the middle. So DNA is double-stranded. This is double-stranded. And RNA is typically single-stranded. And this helps because we don't want RNA to last too long. So this gives it kind of a short life. It's less stable. Whereas the DNA, because it's double-stranded, it's a little bit more stable. More durable. It's also found in the nucleus. So we typically find DNA. DNA is typically in the nucleus only, unless it's the cell cycle. RNA is found in the cytoplasm, mostly. Obviously, it has to come from the nucleus, so it's found in the nucleus, but it works in the cytoplasm, right? DNA typically functions in the nucleus. Now, the two strands of DNA you need to know are held together by none other than hydrogen bonds. So there must be polar pieces within the nucleic acid that are causing it to, that are holding it, or I guess that are causing slightly negative and slightly positive charges, and there's hydrogens in there. And what's happening is that the slightly positive hydrogens are being attracted to the slightly negative, usually oxygens, that are on the other amino acid, on the other strand, on the other piece. So the amino acids that are on this side are going to be attracted to the amino acids on this strand. So the nucleic acids that are on this pink strand are going to be attracted to the amino acids on this other blue strand here. And they're attracted because, let's say that if there's a partial positive charge on the nucleic acid on the pink strand, then there will be a partially negative charge on the nucleic acid on the blue strand, and so those bonds are hydrogen bonds. Hydrogen bonds. Now we mentioned that nucleotides are the building blocks for nucleic acids. And so RNA and DNA have 3 amino acids in common: cytosine, guanine, and adenine. Or you probably know them as just CGA, right? CGA. Now, RNA has a fourth nucleic acid called uracil, or we just call it U. So, oopsies. Uracil is U. Whereas DNA has thymine as its fourth nucleic acid, and we just call it T. So the nucleic acids on RNA are C, G, A, and U, but the nucleic acids in DNA are C, G, A, and T. Okay, so a slight difference. U and T are very similar. They're basically cousins, but they are different from each other. Okay, let's talk a little bit about the structure of a nucleic acid. So the structure of a nucleic acid is very predictable. Now, every nucleic acid has at its center a 5-carbon sugar. Remember I said that we use these pentagons to represent a 5-carbon sugar? There's a 5-carbon sugar. Every nucleic acid has a phosphate group. Okay, so here attached to the sugar is a phosphate group. Remember, a phosphate group is PO4 3-. So it carries overall this negative charge. There's like a negative charge on these phosphate groups. And then it will carry one of 4 of those bases. One of 4 of those bases. This base could be C, it could be G, it could be A, it could be T if it's DNA, or U if it's RNA. And we call this a nitrogenous base because it's full of nitrogens, right? Nitrogenous means that it's full of nitrogen atoms. Now, in DNA, this 5-carbon sugar is deoxyribose. That's in DNA, and that's what the D stands for. The D stands for deoxyribose. In RNA, this 5-carbon sugar is just ribose, and that's what the R in RNA stands for. And then, like I said, between the bases, we've got hydrogen bonds holding those bases together in DNA. RNA doesn't always do this. It does pair like this. And we call this a base pair. So these bases, the base group, the nitrogenous bases of the nucleotides, they like to make base pairs. They like to make base pairs, and they do that with hydrogen bonds. They do that with hydrogen bonds. Now there's one other molecule that we want to just talk a little bit about, and that's ATP. ATP is called adenosine triphosphate. And this is the cellular energy. This is usable cellular energy. Okay, so all cells will use ATP as their energy source. It's a high energy molecule. The triphosphate means that there are 3 phosphates. There are 3 PO4 3- groups. So here you can see them right here. Let's do this. Got 1 here, 2, and 3. We got 3 phosphate groups. And the covalent bonds that hold them together contain energy. They're held together, they store energy. And the third bond here, this chemical bond here, is a high energy bond. Energy bond. Okay, and so when we break that bond, when we break this bond, then what happens is we get free energy. And so our cells will use that free energy to do work, right? The cells can use this energy to do work. Okay? And so it's really important that our cells are able to cycle these two molecules between each other. So what will often happen is that ATP will get used and we get ADP, adenosine diphosphate, out of that. What happens is two things are released. Something called an inorganic phosphate. This is called an inorganic phosphate. And our cells have lots. There's plenty of inorganic phosphate in our cells. Okay, and the other thing that it does is it releases energy. Okay, that's that free energy that we talked about. Now our cells in our mitochondria and in the chloroplasts will put this phosphate, this inorganic phosphate goes back onto an ADP, and we can recycle ADP and change it back into ATP. And of course, this requires energy going in. So we need energy going in, right? And usually this either comes from one of two places. Either in plants, it'll be sunlight, or in animals, this will come from our food. Okay, we'll use that energy. And we talked about how sugars, glucose, are the energy for making ATP. So that's kind of that process there. Okay, so ATP cycles pretty much endlessly with ADP to make energy or to store energy so that our cells can use it. And again, it's very similar to a nucleotide. You can see here that we've got our sugar. So here's our sugar. Here's the 5-carbon sugar, right? Our 5-carbon sugar. Here's the phosphates, right? We've got the phosphate groups over here, right? Those are attached to the sugar. And then over here, here's adenine, which is a nitrogenous base. So adenosine triphosphate, really, this is just a modified nucleic acid. Okay? Right on. So that is the end of our biological molecules chat. And if you have any questions, please let me know.